Hello? Yeah. Oops. So how do I get it back? Just go. Okay. Thank you. Good afternoon, all of you. Thank you very much, Dr. Deepthi, for the introduction. It's a pleasure to come back to INDICON. I was there in the last INDICON meet in Guwahati. What I'm going to speak on today, I think I will have about half an hour, 35. Yeah, so total 45 minutes. Okay. So what I'm going to speak on is are some of the results that we have in artificial intelligence in healthcare. Not only our results, but what is typically going on in this domain. Of course, it's a very, very broad domain, and it's not possible to cover all the aspects. But I'll just give you a feel of a few things where artificial intelligence is playing a major role, few areas of healthcare research where AI has had some successes. So as we all know very quickly, artificial intelligence versus natural intelligence. And in AI, we are trying to make the machine think and act like humans. So that is what we would like to do, but it's now also becoming a matter of contention whether in this era of big data, it's enough to make the machine think or react like a human being, or the machine is in fact going beyond that. And possibly with the plethora of data, that is what is happening on many things where a lot of data needs to be sifted through and conclusions reached. There, the machine is certainly doing better, but the areas where we all know, areas where machines are yet to catch up, will take a very, very long time, if at all, are these aspects of creativity, the common sense, intuition, the sixth sense that human beings have. It's perception, the context, looking at the context and reacting differently to different contexts. Computers are also able to look at context and take different actions, but not as much as we are able to do. So, this, whether a machine can be truly called intelligent, we all know that there is this Turing test, devised long back, 1950s by Alan Turing, where there is an interrogator who cannot look at, there is a computer and a human being. The interrogator cannot see who is responding, which is the computer and which is the human, and is asking some questions, and if the responses are all written, typed, and the Turing test would be passed if at most 30% of the time the machine is mistaken for a human in a 5-minute keyboard conversation. So that was typically what the Turing test demanded. And it was said that in 2014, a computer AI passed this Turing test, not exactly in the way it was defined, but it was able to fool 33% of the judges into thinking that the responder was a human being. So this discussion continues, when to say that a machine is truly intelligent, whether we want a machine to be autonomously intelligent or not, that is also a concern. What if it is left on its own devices with a lot of intelligence built in, or with a lot of autonomy built in, then how it will behave? These are all concerns which continue, the ethical part, the explainable part of AI, etc. The CAPTCHA is a reverse Turing test. We do it all the time these days, where the computer is the interrogator, not the human. The test is administered by the computer, and it has to determine whether the responder is a human being or a computer, a machine, a bot or something. So that's the reverse Turing test that we are these days doing all the time. So quickly, we know that artificial intelligence in itself is a very big domain. A part of it is machine learning, where some learning goes on. There are many AI tasks which are not machine learning. For example, the expert system is not really machine learning. And within machine learning, we have deep learning. It's mentioned separately because these days deep learning has become so prevalent in everything that we are doing. We are going towards deep learning. So machine learning typically where you are able to learn something which is not given as an example. So to be able to infer something about unknown situations, that's where the machine learns. So for example, just a database query retrieval is certainly not machine learning, but if a computer is shown several images of cats and dogs and a new image, something that it has not seen before, is presented to it and it is able to infer that this is either a cat or a dog, so that is where the machine learning is coming in. Now, deep learning, as we said, goes further into machine learning. It's an advanced version of machine learning, and it has become important because of the huge computational power that we have at our disposal these days, and because of the huge amount of data processing capabilities, the data storage also, and the huge amount of data which is getting collected. So we can see that as and when the amount of data increases compared to other algorithms, deep learning starts performing much better. For small data, it doesn't really matter. Maybe deep learning is an overkill in those situations. So we'll come to that shortly. But as I said, deep learning, these are machine learning techniques primarily based on artificial neural networks. And main task of deep learning models is feature engineering. Features which in pre-deep learning days, we would extract and present to the machine to learn to classify, to learn to cluster or whatever. That part is taken care of by the deep learning models, which learns the representation of the data set. So that's representation learning is what is the key thing in deep learning. And there are many different architectures which we can think of and which we know that they exist. And about the applications of AI, I don't think this audience needs that. It's sort of omnipresent these days. Wherever you, whichever domain you want to think of, you have applications of AI there. The self-driving cars, agriculture, healthcare, retail, industrial automation, almost very few areas of life left which is not touched by artificial intelligence. To come to healthcare research now, what's the definition? The definition is efforts made to maintain or restore physical, mental, or emotional well-being, especially by trained and licensed professionals. That's the definition of healthcare. Now we want to bring in automation there, we want to do AI there. So healthcare research plus data science, where the data drives the science. The amount of data that we have, it tells what are the principles which have given rise to that data. So data science, data analytics in healthcare, we can broadly classify as descriptive, which just describes what happened. It could be predictive, where we predict what is going to happen. Prescriptive, it prescribes what should happen, what should be done, and also personalized. So what should be done for me in particular? Because what should be done in a particular situation would depend on how I am made up. So that is where personalization is coming in a big way these days. Revolutions in healthcare have been many. First, of course, when the structure of DNA was discovered, the double helical structure, 1953, then we were able to look deeper into the molecules inside the cells. Then came the era of genomics, when we were able to look at the entire genomes and understand the genetic basis of diseases. Of course, thereafter we moved on to systems biology, where you don't look at individual molecules, but you look at how they interact at the systems level, how if you do something here, how it affects something somewhere else. So that's at the systems biology level research started. And the convergence happened in recent times where life sciences, engineering, mathematics, statistics, physical sciences all came together. That is this era where we are, where it is an era of convergence, big data and convergence. So all the different disciplines come together to solve pertinent, important problems. So as I said, we will be looking at big and complex data in healthcare at different levels, because healthcare data are of many different types. For example, these could be sequences, these could be graph data, because of the interactions which happen between the different types of molecules which are there. Clinical information, that's also healthcare data. The doctor writes down the clinical information, age, gender, blood pressure, so and so, the height, weight, everything, all together, plus the different parameters that a person, typically that we present when we go to a doctor. These all come under the clinical information, that mining that, making sense out of that, extracting useful information from that, that's also an important part of healthcare research. Epidemiological studies, socioeconomic analysis, it's nothing to do with the biology part. Socioeconomic analysis, health economics, these are also important components of healthcare research. So, as I said, Human Genome Project, understanding the genome was one of the core problems which was handled between 1999 to 2013. And it was also very interesting because that Human Genome Project required a lot of computational methods to solve the problem. So essentially what was there was a long genome. So just to go into what a genome is, if we look at the cell, inside the cell we have the nucleus, inside the nucleus we have the chromosomes. Each chromosome, if you look, it's a double-stranded DNA, it's going like this. If we take out one strand and take out one strand from each of the chromosomes that are there inside the cell, lay it side by side, it's a very long string of just 4 characters, which are A, T, C, and G, and that is the genome. Understanding what that long string is made up of is the understanding the genome, essentially sequencing the genome. And that was done for the first time way back, 1990, 2013, around that time. So, but then technology existed where you could only sequence small pieces, not such a big string. You could only sequence small pieces. So therefore, you had to cut the long strand, let's say, the long strand into smaller, shorter pieces. You could then sequence these shorter pieces, but the challenge was how to rebuild the long string. So that is where computational methods became important, and without that it could not have been solved. So essentially how it was solved was multiple copies, not just a single string, but multiple copies were made of that string, and then each was cut into shorter pieces and sequenced. And now what you would do essentially is, because there are multiple copies, for the same portion there will be multiple presents in the different shorter sequences, and then you start doing alignment of the sequences. So you start fragment by fragment and see which fragments match, and then the hanging portion, and then what other fragments match that hanging portion, and in that way you try to rebuild the big genome. That was the fragment assembly problem, typically a mathematical or a computational problem. But unless these two came together, the genome project would not have succeeded. So that was a very interesting application, a real-life problem, which was solved using such methods. So let me now come to some of the work that we've been doing in our lab. Let me tell you a little bit about a small molecule that I have been looking at for some time in our lab. This small molecule is a microRNA. And microRNA, this molecule, is actually heavily implicated in different types of cancer. There are, in almost all types of diseases, you can find the involvement of certain microRNAs, but particularly in almost all types of cancer, there are microRNAs which are doing something wrong. Their levels are going here and there. So it's important to understand what the microRNAs are doing in order to be able to understand the disease also. So as you can see here, that long string, let's assume that long string at the top, the long line, is the genome. Okay? There are certain parts of the genome which are the genes, which are genes in the sense they code for certain proteins, usually. Usually genes would code for certain proteins. So you can see, for example, this side, there is a protein coding gene, the blue portion, a protein coding gene. Now, typically this protein coding gene would have formed a molecule normally called the messenger RNA. You can see it here, the mRNA. And this mRNA would have produced some protein. Okay, so that is the normal pathway for a gene. Now what happens is, at a particular point of time, because of certain reasons, another gene, which is a microRNA gene, that gets active, that becomes active, and produces a small RNA molecule, which is called the microRNA. And you can see on that side, on the right-hand side, you can see that a microRNA has got produced. And what it does, essentially, this microRNA, it will find its target in one of the messenger RNAs which are out there in the cellular environment. And it will go, that microRNA will go and either bind to this messenger RNA, just sit tightly with the messenger RNA, or it will degrade, it will sort of cut the messenger RNA. So either that messenger RNA degrades, disappears, or it is just repressed because something else has come and has bound itself to that messenger RNA. So either way, the end result is that the messenger RNA, the protein which it would have produced, normally does not get produced. So why is this a problem? Now, think of different situations where the protein is important. For example, normally human cells divide, and therefore human cells also have to be killed. So that's called apoptosis, cell killing. Now, there are proteins which are important for killing of cells. Suppose this messenger RNA was responsible for production of such a protein which was responsible for cell killing. Now, once the microRNA gets active, which targets this messenger RNA, then what happens is that protein does not get produced, whose responsibility was, or which was playing a role in cell killing. Therefore, cells are not getting killed. Cells are dividing, but cells are not getting killed. So that essentially is an explosion of cells in that region, leading to cancer. That is what we know, that's cancer. So that microRNA did something wrong, because of which cancer appeared. That is a very typical situation. That is happening almost in all the cancers that we have. We see that there are microRNAs which are heavily involved, which seem to be a culprit in the diseases. There might be many other, that's not the only one. There might be many other pathways by which that cancer is appearing, but this is also one of the ways in which gives rise to cancer. So that is what we have been studying for some time now. So therefore, it is important, you see, to be able to tell, for example, which microRNA will target which messenger RNA. So if we just go deeper into this, essentially it looks like a classification problem. We have, let's say, a set of microRNA, mRNA pairs where we know that the microRNA is targeting the messenger RNA. We have a set of such data, set of such examples. We also, let's say, have a set of examples of microRNA which do not target the corresponding mRNA. So these are all biologically validated data. Let's assume we have such data, and those are the non-targets. So now we have a set of examples where we have examples of targets and non-targets. So this, as you can appreciate, becomes a typical classification problem. We have training data, we can train, and then given an unknown pair of microRNA and mRNA, we try to predict whether the microRNA will target the messenger RNA or not. What is the problem here? That seems to be a simple enough issue. The problem is that while there are a lot of positive data, that is targets, validated targets, there are very few validated non-targets. Because when the biologist does an experiment and finds a target, the biologist reports it. But when it doesn't work out, it is not, the person was thinking that, okay, this might target this microRNA, might target this messenger RNA, then, and it didn't work out, that is usually not reported. So those negative data, there was a lack of negative data. And that is what we realized when we were doing the literature survey, looking at the different methods which are out there and trying to compare. We saw that most of the methods, their results were widely varying. So then we went deeper into the problem, where was the issue, and then we realized that in many such classification tasks where the negative data was missing, what the researcher did was artificially generated negative data. And this artificially generated negative data became very different from the positive data. So during training, classification performance was good, and then during testing, it deteriorated. Because those classifiers had poor generalization capabilities. So what we did is we tackled this problem, not really the classification part of the problem, but we tackled the problem of this remarkable lack of negative data using a method which I will not go into detail of that method, but we did we came up with a protocol where we were able to actually identify very possible negative data, in the sense high confident pairs, which we were almost certain would be negative. But these are not validated. These are not biologically validated. Nobody has experimented and told me that, because if there is a biological validation, then we know that's a negative data. But what we did here was we took the union of several different prediction algorithms. We took positive data, and then from there we tried to filter out through different mechanisms any pair which had a remote possibility of being a true positive. So from a set of predicted positives, we filtered out a large set which had even a remote possibility of being a true positive. What was left with was a smaller set of data which is predicted to be positive, meaning it would target, but has very low chance of being true positive. So that is what we took as the negative data. That means our negative data now looked more like the positive data. And this particular work got importance not because of the classification that was happening, but because of that negative data selection approach. Many researchers, we later found, were actually then looking at that negative data creation part and adopting that negative data creation protocol, and then doing whatever classifier they were fond of. So our method was called target miner. We of course compared as usual with many other methods, and this plot shows, you see on the x-axis we have the false positive rate, and on the y-axis we have the true positive rate. Now what we would like a classifier, where would we like a classifier to belong? In the quadrant number 2, which is number 2, because that has a lower false positive rates and higher true positive rates. And true enough, we found our method performing there, along with another method which is also very popular, target scan, while most other methods either were too optimistic or too pessimistic. So that was our one of the works which we did. And then we built on this work. Not only microRNA, we actually now plugged in different other molecules also, along with the microRNA. So you see, there's an interesting thing to note here, that I talked about the genome. So if I take a cell from the skin here, if I take a cell from some other tissue, maybe the heart or the lungs or anywhere, and we look at the genome, meaning the long strand of DNA, it's the same. Okay, so that sequence A, T, T, C, A, G, T, whatever it is, that is the same wherever we take the genome from. Then the question comes that if the genome everywhere is the same, then why are the cells behaving differently? The cells are behaving differently primarily because even though the genomes are the same, what does that mean? The genomes being the same means the set of genes is the same. So all the cells should have the same set of genes, and therefore the same set of genes should produce the same set of or similar set of proteins. That is not happening because a gene will only express itself, will only produce its protein if there is a switch for each gene. If the switch is on, only then will the gene express itself, otherwise not. And what is that switch that turns on or turns off a gene? That switch also is a protein. And if the switch is a protein, that means it is being produced from some gene in that same genome. So, if you think of it a little more in deep, then you will find that there is a very complicated network going on here. So, one gene producing a protein which controls another gene, and this gene might produce another protein which will control some other gene, etc. So, there is a gene regulatory network. This is called a gene regulatory network in action in the cell, right? Now, earlier only this gene regulatory network was being considered. What we did was, we thought, well, because this microRNA is, because when we started working on microRNAs, not much work was out there. So, we noticed that this microRNA is also controlling the expression of its target messenger RNAs, may not be directly the genes, but the product of the genes. Messenger RNA is a product of a gene. So, gene, messenger RNA, and then the protein. So, if we control the messenger RNA, then that in effect is a control on the gene itself. So, what we did was, we built in that gene regulatory network the effect of microRNAs. So, one more molecule increased in that gene regulatory network. So, you will find here, there are certain microRNAs in this network. Well, not really. The network it would look different. So, there were microRNAs, there were genes, and there were transcription factors. So, now we analyzed this entire network and then did a DFS level method, somebody else's method, and then we looked at how it came out. And it was very surprising to see that it came into, we could find a 3-layer type of hierarchical structure there. And that appeared across networks. We were looking at this breast cancer specific network, so for this particular example. So we found that at the top, all those 5 molecules are microRNAs. They just happen to be microRNAs for this breast cancer specific network. And when we looked at the literature regarding those microRNAs, what we noted was, out of those 5, 3 already had sufficient information in the literature that they were onco-MIRs, onco-microRNAs, meaning that they were responsible for cancer. But for 2, there was not any information actually. So, microRNA 155 and microRNA 202 turned out that there was no information available in the literature. So, these actually, what we did was, that's where we published, because after that we did not have the expertise to go on. Later we found that one of those microRNAs were validated independently, which is sort of a good thing. So, this is one way in which we were able to identify, to predict certain markers. These are markers. So, this microRNA is a potential marker for breast cancer, meaning that if a patient presents herself and the microRNA expressions are looked into, it is microRNA 155 is a potential microRNA whose expression should be looked into to infer something about the disease. This certainly will not be, a complete inference, but it will give some idea. So, that's how actually these biological markers are usually predicted, are usually identified, and this is one such work we did. We also did another work in colorectal specific cancer. Again, similar results came out, where at the top level, we had actually 3 microRNAs and 2 transcription factors. So what we found is out of those 3 microRNAs, 2 had enough literature evidence, 1 did not. So this is again one prediction for a biomarker for colorectal cancer. So with that, let me move on to another quickly. I don't think I have much time. So convolutional neural networks, I think nowadays almost all of us know about it or have heard about it. It goes through, as you can see here, some layers of convolution and pooling, and then it comes down in size from the original image. Let's say image is the input. If image is the input, then that's how it goes. But the general principle is the same, whatever is the input. And at the end, we have a flattening of the data, and that data goes into a classification system. Typically, maybe it could be a multi-layered perceptron, an artificial neural network, which does the classification. But the important, interesting thing here is, you see, earlier, when we were doing image processing, we had to extract features from the image and then give it to some classifier, which is a favorite classifier of some researcher. It could be naive Bayes classifier, it could be SVM, it could be a KNN classifier, whatever, but we have to extract the features. Here, we are directly giving the images. We don't bother what features are important, what features are not important, and it's through these steps of convolution and pooling that the representation of the image gets learned. So that is where the challenge is, because you see, when you are doing the convolution, for example, it's through a sort of a kernel with which you are convolving the image. That kernel parameters, those need to be learned, and there are multiple layers of that. Then you come to the multi-layered perceptron, the weights, there are weights there which have to be learned. So millions of parameters which have to be learned in order to do something good out of it. And for learning millions of parameters, you need humongous amount of training data. And for both of these things, learning so many parameters and so much of data handling was not possible in earlier times. Otherwise, there's nothing much new here, but it was not possible. It was, you could only have a few layers of a neural network, for example, and that's it, because even then learning was difficult. But now, since the computational power has gone up, since storage capacity has gone up, so therefore this has become possible. And this sort of gave a paradigm shift. Although it has a lot of problems, it has a lot of trouble, yet it has given so remarkable classification results that most researchers have now moved into deep learning for a long time now. So, to quickly give those who are new to this area, like, what are this convolution doing? For example, it's learning it's like a edge filter. If this is the image and you have a horizontal edge filter, then what do you want typically learn? You see, the vertical parts are gone. You are left with the horizontal features. Similarly, if you look at this and you convolve it with a filter, with a kernel which looks at the vertical, it's a vertical edge filter, then mostly you will see that the vertical lines have appeared. So that is how, through these combinations, gradually the representation is getting learned. So, and then some developments happened. This is not our work, but a group close to ours, their work, where they came up with something called fuzzy at-risk convolutions. I will not go into the details of that. But essentially, thereafter, it was applied to COVID X-ray images of the lungs and the chest X-ray images. And you can see certain negative samples here. Those are positive samples. So it was classified using fuzzy at-risk CNN. And there is this method called GradCam visualization, which not only gives the classification, but through GradCam visualization, you can see which portions were important for the classification. So this is sort of going towards a type of explainability, trying to explain that, okay, I got this as class 1, but what was important in that image which gave this result of class 1? Okay? So here it was seen that the CNN was able to capture to a large extent relevant portions of the image for doing its classification. Now, you might have heard of this strange type of result where the CNN was trained to identify polar bear, for example. Okay? And it did fantastically well. And then it was shown just a mountain with snow, and it said polar bear. So then it was realized that it had not actually learned the bear. It had learned the white snow, because all the polar bears, the pictures were in the snow, and therefore the white color was learned. It was not really the polar bear that the system had learned. So that is why it is important to see what it is learning, whether it's learning something meaningful or not. This is model performance, of course. The first one was the proposed method by our collaborators, and accuracy also we can see it's very good, it's quite good, while the parameter count, because of the innovation brought in, the number of parameters, which is typically, you can see in some methods it's 25 million, 130 million parameters to be learned, it was of the order of 0.062 million parameters to be learned. Some more results with some more innovations built in. Why is this not moving? Oop. It refuses to move ahead of this. Anybody? Okay, it's become very slow. What is happening? No, it's not working as desired. Okay. So again, create some more work which was done in this area, and just some results that this is for an MRI data set analysis. As you can see, at the top is the input, the segmentation result of the proposed model in the middle row, and the last row is the ground truth. And the middle row actually shows the ground truth superposed with the result which came. So whatever is yellow is matching with the ground truth. The red portions or the blue portions, etc., those are which are not matching with the ground truth. So you can see it's mostly matching with the ground truth here. This is using, of course, some multi-resolution images, and okay. While when it was compared with many other methods which are out there, with many other like UNet, UNet++, etc., then you can see that the previous one was doing much better because it matched the ground truth to a very high extent. But these, they have problems. We can see it's not matching the ground truth to the desired extent. Forget about this result. Quickly, because I don't have time, I'll just tell you that there is another work which we are interested in, we've been doing, which is in graph neural networks. And what we are doing here is actually looking at drug-drug interaction network, because drugs interact with each other, and there are databases which tell us which drugs interact with which drugs. There are gene-gene interaction networks. There are diseases which interact, there are multiple diseases out there, and pathway interaction network. And all these together, we built into this concept called graph neural networks, where it's a graph embedding is learned. And we want to, actually this is an ongoing work, what we want to do is to sort of answer queries like, does some drug treat or can be used to treat some particular disease? Is gene, meaning some gene, responsible for some disease, etc. By doing this, this graph neural network based analysis, we are trying to respond to queries of that type. Plus, it could be useful for example, you are taking 3 different medicines, okay? And you have certain other symptoms, you've gone to the doctor. Using systems like this, when the doctor is trying to prescribe you a new medicine, the doctor needs to know what are the potential adverse effects that might happen, adverse drug effects that might happen because of other medicines that you are already taking. And that adverse drug effects, that's ADE, that is not completely understood. It's not, in fact, it's not understood very poorly. What drugs would interact with what drugs in presence of which symptoms and in presence of certain genetic predisposition of that individual. So altogether, trying to predict adverse drug effects. This is one work which we've recently got a project, and we are working on answering queries like that. And I will quickly conclude with one last by describing one work which we did several years back, which is in the area of drug design. Okay, so I just wanted to give a broad perspective of different types of work that we've been doing in the AI healthcare domain. And here, so it's typically said that drugs will no longer be will nowadays be designed on computers. It will not be just discovered from natural systems, but it will be designed on computers. And one drug molecule, this is called the drug Relenza for flu, that was designed using principles of computer-based drug designs, rational drug designs. So what we did was, we actually looked at the drug design problem as an optimization problem. So because when a drug works, what happens is a small molecule, the drug molecule, it targets certain proteins which are misbehaving in the system, which are appearing in the disease pathways. And how do we do this? How does the small molecule, the drug molecule, go and interact with the protein? Well, the protein actually has certain active sites in the surface of the protein where the action takes place. So suppose there is one cavity here where because of this binding with something else, a disease is appearing. Now, if this small drug molecule can go and sit here before that harmful binding takes place, then you have a hope of arresting that disease. And that's typically how most of the drugs work. They target certain proteins and certain active sites of the proteins. And that is what we did. We designed small molecules using our method, which is an optimization. What we were trying to do is this molecule should have properties such that the interaction of this molecule with its target protein, the internal energy of this molecule, all of these would be minimized. And because we know that when these energies are minimized, then the system is stable. So we were looking at optimization methods, and we actually posed the problem of drug design as a multi-objective optimization technique. These were the objectives which we had taken to optimize. And quickly some results. We were targeting a molecule called RecA of Mycobacterium tuberculosis. And you can see here this green ribbon-like structure, that is the RecA protein. RecA typically, what it does is, this is the protein of the bacteria. And when the bacteria attacks, then typically what we do is, we take antibiotics, which damages the DNA. RecA is a protein of the bacteria whose task is to repair the DNA. So that antibiotic then, after a little while, loses its efficacy because whatever DNA is getting damaged, the bacteria is repairing itself. So if that RecA protein can be targeted by drug molecules, then we hope that the repair of the DNA would not happen, and that is how, that could be one way of looking at that, or doing something about the disease. So we were trying to design small molecules, typically which look like this, which would go and fit into the active site of the RecA target. And we were able to design some molecules which appeared in the database of already known inhibitors of RecA. So there is a database which already gives that, okay, these are the molecules which are known to inhibit RecA. And some of those molecules were regenerated by our method, giving us more confidence on the method. But at the same time, some other molecules which are not appearing in that database also got generated. So this could be possible or probable binders, probable lead molecules. So a lead molecule is not a drug molecule. Okay? A lead molecule is only something which is a worthy starting point. And then you have to see what this lead molecule actually leads to, because often what happens is when what happened? Oh, I'm going. What to do? Yeah, just go back, but that's it. You can just. So what happens is these small lead molecules, often it has to go through a lot of testing before it can actually reach the market, and that is a very long drawn process. Most of the times these drugs are discarded because the side effects are very severe. So the reason for side effects appearing for any drug, for that matter, whatever drug you can think of, it will have some side effects. And the side effect is because this drug molecule, while it is very nicely able to bind to its target sites, it also goes and binds into some non-target sites. So it's not very specific binding. The more specific the binding is, that it only goes and binds to its target and binds nowhere else, then that is the best thing. But that doesn't happen. And that is why the side effects manifest themselves. And if the side effects are so serious that the efficacy, the side effect is worse, so bad that you cannot have that drug, then of course you have to discard that. So most of the lead molecules people start, but most of them actually do not reach the stage where they can come to the market. Most of them are discarded on the way because of various different factors. So what is important yet is to be able to constantly be at it, constantly look at new and new possible lead molecules, and that's what one of the benefits of this work was. We again published up to this level, did some QSR studies, meaning we did some predictions of the activity of these molecules with respect to that target. There are methods called QSAR, quantitative structure activity relationships, which we did. Those studies we did, found good results, and that's where we stopped. So that was another work which I thought I will just tell you. Another aspect of healthcare research in drug design that could be done. Of course, the opportunities these days are immense because of the huge computational power, the huge storage capacities, etc. But then demand for high intellectual capabilities, high intellectual prowess is also very important, where I think we should, from India, we have a very big role to play here, because we are good at it. Good at programming and things like that, and mathematics, statistical models, etc. So the challenges, of course, remain. We all know that, researchers, that there is this government, academia, industry disconnect which we need to bridge. So I will not spend much time on it. Of course, we have this academia where we have more time, more appetite for risk. We can afford to fail. But industry, of course, strict timelines and less appetite for risk, etc. So a good connect, along with the government coming in with suitable interventions, with suitable fundings also. Funding is one great problem in our country, because the amount we spend on research is very minimal. It has to go up. industry funding, government funding, both have to increase. So those are the challenges that we continue facing today. With that, I will conclude because I think I have run well beyond my allotted time. My acknowledgements to the different funding agencies, my institute, etc., and of course to all my students. I have one former PhD student who is sitting right here, and she has done great work in multi-objective and multi-objective optimization, Mona Lisa Pal. With that, thank you very much. There is some problem here. It doesn't want to work. So thank you very much. If you have any one or two questions, I'll be happy to take them.
It was great to listen every time you, ma'am, as it is like that. I just want to know, this is the curiosity. It is good that you are giving responsibility to machine for this artificial intelligence to take care of humanity in terms of medicine and disease detection and all. What will happen if intelligence will take over humanity? Like the sci-fi movies, if we refer, the sci-fi movies, if we refer like Age of Ultron or something kind of thing, what is the chances of that thing to happen, or is there any possibility to take over that?
Well, I don't know in the future what is going to happen where, because life has changed dramatically. So a small smartphone has changed the way we think, way we behave, way we react, way we act, everything. But we are not giving the responsibility to the machine. The control is still in the hands of the human. We are using the machine to assist, to help, to bring up some information, after which it's the human being which will actually look at that. As of now, at least, that is what is happening, that the entire responsibility is not the machine. It's a collaboration between the machine and the human, and we hope that is how it is going to stay. That it will remain a collaboration where the human is in charge, uses the capacity, the capabilities of the machine to, look at all possibilities, to look at like in a chess playing, but it's all very methodical there. But in such machine learning areas, it will come up with something new, but till now, I don't think there is, in the near future, that apprehension of machine controlling our lives, other than what machine is already controlling our lives in the sense of how we think, and all the kids all the time looking at only at, even when the friends are sitting side by side, they're looking at their mobiles. So that's already a level of control of machines on our lives, but not in the way maybe you are thinking. I don't see it happening, at least sometime soon. There might be other people here who might have different views, but I think it is going to be a healthy collaboration, and where the machine is used by the human to get to extract its benefits.
Yeah, thank you so much. Actually, I referred giving responsibility to machine because in underwater communication, when bots are sent, one bot is actually given responsibility to take care of the other bot to make the networks, because communication is a challenging there. So actually I referred that one, so giving responsibility to that bot. If something is happening and taking the responsibility of the other bots, that is doing. So in that terms actually I wanted to say. Thank you so much.
Sure.
Thank you, ma'am. Actually, my question is from DNA, genome sequencing.
Huh?
My question is from genome DNA sequencing. Like you told earlier that there are 4 chemical elements, ATGC, right?
Huh.
In the DNA. So how we are differentiating among the humans, like this having different DNA or this that is having different DNA?
How we are distinguishing between?
Two different DNAs, like they are based on the length of DNA. That's why we are able to distinguish between DNA of two persons, or like that.
Yeah, so essentially if we take DNA of your two human beings, it will be similar to a very large extent, in the sense when you align those those sequences, they will generally be matching in these in their locations, but there will be a few differences. That's why the individuals are different.
Actually, I have also used the DNA genome sequence for hardware security purpose, and I am researching on that field.
For hardware security.
Like hardware coprocessors, I am securing by the DNA sequence of human being. But in that, I have defined some certain constraints for security purpose. But in general, in biological manner, how we are differentiating the two DNAs of a person?
That's what I said. I mean, you match those DNAs and find out the points of divergence where they are not matching. The characters are not matching. So when you have, for example, if you have a set of DNA sequences, again you find homology search, you align them like this, wherever the matching score, there is a matching score which you can compute. If it is a straight matching, one score. If there are mismatches, then the different types of A here and a T here, or a C here and a T here, the mismatch score would be defined differently. There are like insertions. So it's, for example, there is one DNA, and the other is essentially that DNA, but something has got inserted in the middle. So there are indels or insertions, deletions, etc.
So for matching, the DNA length should be higher. If the length is, let's say, smaller and is having only 4 chemical elements, so it may be a higher probability that the two DNA are matched.
Exactly matching, no. Between two different individuals, exact match will not be there.
Okay, thank you.
There will be differences.
Thank you.
Thank you very much.
Thank you, ma'am, for this exhaustive session. I call upon Professor Saha to hand over a token of a memento as a token of our appreciation.